Good evening, student. Today we are going to learn asexual reproduction in flowering plants. What is the meaning of reproduction? Reproduction means producing a new offspring which will be the same in physical character and genetical character is known as reproductions. It is more reproduction mainly two types, sexual and asexual. What is asexual? Asexual reproduction is a mode of reproduction in which a new offspring is produced by a single parent. Means in asexual reproduction, only one parent will be involved, and it is mainly a different types of binary fission, budding, fragmentation, vegetative propagations, sporogenesis. These are types of asexual reproduction. It is mainly found in a grass, runner types, rise in ginger we can say rhizomes, and bulb in we can see in bulb potatoes. And what is sexual reproduction? Sexual reproduction means haploid gametes which will come from one haploid gamete will come from the male and one haploid gamete will come from the females, and it will form a diploid zygote, so which is known as sexual reproductions. Professor P. Maheshwari, he is known as father of Indian plant embryology. So, sexual reproduction in flowering plants means what is flowers? Flowering plants, what is flowering plants? All angiosperm is known as flowering plants. They produce a flowers. Whatever plants will produce a flowers is known as flowering plants. So, flower is a very important for reproductions. So, we'll discuss about it. So, what is flower? It is a modified shoot parts. So here we can see in flowers the nodes and internodes will be present. There are four internodes will be present. These are known as calyx, corolla, androecium, and gynoeciums. Androecium it is a male reproductive organs, and gynoecium it is a female reproductive organs. So it is very important. So we'll discuss about it. First, we'll discuss about the androecium. These are the types of these are diagrams of androeciums. Here we can see a diagrams of androeciums. Um, it will look like it. The upper side, the anther will be present. The bilobed structure we can see here. It is known as anthers. And lower side, the ventral view we can see. Filaments. Lower side is known as filaments. Entire it is known as stamens. It is also known as microsporophyll. Why microsporophyll? The microspore and fill. Fill means filaments. So it is known as microsporophyll. The spores will be present here. On the basis of fixation of anthers with the help of filaments. So it is divided mainly four types: basifix, adnate, dorsifix, and versatile. Next, anther is very important. So what is anther? The long and slender stalk called the filaments, and the terminal generally bilobed structure is called the anthers. Suppose that we'll cut the anther by transverse section. So we will take a transverse section. So it will look like this types. It is known as theca. What is known as theca? In theca, we can see spores. One spores, two spores, three spores, and four spores. On the basis of theca, it is divided into two types: monoethicous and diethicous. What is monoethicous? Suppose that only one theca will be present, and suppose that here it is one theca. Here we can see a spore. Spores. On the basis of spores, it is divided into two types. Suppose that one spores will be present, so it is known as monosporangiate. And suppose that two spores will be present, so it is known as disporangiate. In Arcithobium, example, in Arcithobium we can see a monosporangiate, and disporangiate we can see in Malvaceae and other. And next in Diatheca, Diathecus mostly it Diathecus, it is found in many plants, in different many plants. On the here we can see spores, one spores, two spores, three spores, and four spores. So it is known as tetrasporangiate. It is known as tetrasporangiate. What is microsporogenesis? The formation of microspore from pollen mother cells through the meiosis is called as microsporogenesis. What is megasporogenesis? The process of formation of megaspores from the megaspores mother cells. The MMC is large cells, dense cytoplasm, and prominent nucleus. It undergoes meiosis, resulting in the production of four megaspores, microsporogenesis. Earlier we had discussed about it. We can see at ears, and third, the vascular bundle will be present here. With the help of vascular bundles means xylem phloem will be present here. So with the help of xylem phloem, they supply the nutrients and water to the anthers. And we'll discuss about the structure. So we'll take these parts. Only one these parts will study the outer side. The epidermal cell will be present, epidermis. So it is known as epidermis. Outer side, it protects, it is single thin cells, and below the epidermis the endothelium will be present. It is during the youngs, uh, earlier it will be a single cell. After at the maturation it will be divided into 3 to 4 layers. The outer layer will be thin, and below the outer layer the thick layer will be present because of alpha callose and alpha cellulose and callose. In some place you will see the alpha cellulose and callose will be absent, so which is known as stomium. So because of alpha cellulose and callose will be absent, so it is weaker in weaker parts. So the anther will be break hairs, and the all spores will come out from the hairs. So it is very important, the dehiscence of the anthers. Below the endothelium, the middle layer will be present. So middle layer, it is a it supplies the food to the spores. It is very important. It supplies the food. It is made up of parenchymatous tissues, parenchyma cells. So it is parenchymatous tissues. Here we'll see a function of tapetum. Tapetum is very important for spores. It supplies the nutrition to the developing microspores, MMC or PMC, secretion of enzymes and hormones. It contributes sporulin through UV's body, thus plays an important role in the pollen ball formation. What is sporulin? It is very important. It is made up of biopolymer and phenolic compounds. So it protects the spores from the thermal reaction and chemical reactions. So it is very important. It protect the spores for long times. Formation of UV bodies. The pollen kit material is contributed by tapetal cells and is later transferred to the pollen surface. Exin protein responsible for rejection reaction of the stigma are present in cavities in the exams. These proteins are derived from the it release callus enzymes. So it is very important, and other parts we'll discuss in the next lectures. Thanks.